Hello everybody and welcome back to another BrevWise video. Today we have an IB Biology specimen paper. That's paper 1A, which is a multiple choice paper. We're going to be going through this multiple choice section, just going through it blind, working through each of the solutions as you would, so you guys get a full understanding of how to answer each one. So without further ado, let's get right into it. Question 1. What allows the movement of water under tension in the xylem? So we know water under tension, what allows movement. We're looking for something to do with cohesion between the water molecules, perhaps even adhesion of water molecules to the xylem walls. So adhesion of water molecules to dissolved mineral salts, that's not true. They adhere to the xylem walls. Sorry, let me just get rid of this. Okay, cohesion of water molecules due to hydrogen bonding. That is true. That is how cohesion occurs. It's through hydrogen bonding. Adhesion between water molecules due to uneven sharing of charges. It's a trick because it says adhesion. Adhesion, remember, is not between water molecules. It's between the xylem wall and the water molecules. Cohesion between water molecules and other polar substances. This is again not true. So we know that it's B. Okay. This diagram shows the elements present in two organic molecules, W and X. Which molecules could W and X be? So you can see W contains phosphorus, carbon, oxygen, hydrogen, and so yeah, W contains phosphorus, X contains nitrogen, and they both share these elements in the middle. And yeah, so let's just go through it by looking through each of these options and seeing which ones are plausible. So amino acid does contain nitrogen and the aforementioned. Triglyceride, no. Protein contains amino acids, hence it does contain that. Fatty acid, no. Okay, so we're narrowed down to these two, and now that we think it's between A and B, A and C, we know that monosaccharide won't have this phosphorus, and we know that our phospholipid, it's in the name, phospholipid does contain phosphorus, so yeah, our answer is C. Okay, which property of DNA explains how genetic information can be replicated accurately? Okay, so they're going to give us all properties of DNA, and just we have to look for which one fits this, replicated accurately. So complementary base pairing seems pretty plausible because that explains why only certain base pairing can occur, right? If we have these complementary pairs, it's only going to bind a certain way, so that pair a certain way, so that seems pretty plausible. Double helical shape, while it is a property of DNA, it isn't really relevant in terms of how it's replicated accurately. So 5' to 3' bonding in sugar phosphate backbone, again, it's a property of DNA, but yeah, not really relevant in this case. And binding with histones, no, that's more to do with the DNA packing, how and transcription regulation, So I think it's pretty safe to say our answer is A. Question number 4, we have which molecules are produced during the hydrolysis of the triglyceride molecule. Okay, triglycerides, so we know we have our glycerol and then our 3 hydrocarbon chains, right? So we have water and glycerol, that's not true. Water and fatty acids, not true. Water and lipids, not true. It's fatty acids and glycerol. Yeah. The structure of monomers affects the structure and function of the polymers they form. Which row describes the structural features of polysaccharides made from alpha glucose and beta glucose? So monomer says, okay, so it's saying that these are the monomers and these are the polymers they produce. This is the shape of the polymer. Okay, so A is saying that the monomers are alpha glucose and beta glucose, forming starch and cellulose, and the shape of the polymer is unbranched straight, branched helical. The monomers in this case are correct. Alpha glucose is responsible for starch, beta glucose for cellulose, but remember that starch is branched. Starch is a branched molecule, and cellulose is straight. It has a structural role, and because of the way the beta glucose bonds, because the next glucose has to invert itself, it's going to be in a straight line. There's no kinks, whereas the starch has 1 to 4 glycosidic bonds and 1,6 glycosidic bonds. Okay, So B gives us the same alpha glucose, beta glucose. Actually, not no. D and C has swapped them the other way around, so we know these two are wrong. And B gives us alpha glucose starch, beta glucose cellulose. Starch is branched, helical. Cellulose is unbranched, straight. So we know that B is correct. The diagram shows structure of insulin. From the diagram, what can be concluded from the structure of insulin? Okay, so make sure we pay attention to the diagram so we see this amino acid sequence, and we are seeing, what can we see? So this is two different chains. So this is going to be, you can see this is sulfur, right? And maybe you haven't seen it in person, but you've definitely heard the term disulfide bonds. This is what you call a disulfide bond, occurring between the cysteine residues. So you can see that's where they occur from. Yeah, no need to know that, but just know that these are disulfide bonds associated with a tertiary structure. So, okay, from the diagram, what can be concluded? It is composed of two polypeptide chains. We have two, stabilized by disulfide bonds. That seems very plausible. That's what we interpreted before seeing the options, but let's see. It's simple protein composed of one polypeptide chain. We just simply know that's not true. We see two polypeptide chains. Fibrous protein, that's also not evident here. And its molecules do not display quaternary structure. That is, again, not true. It does display, quaternary structure because we have these two polypeptide chains. Great. Okay. Question 7. The diagram represents the metabolic pathway that converts threonine to isoleucine. Remember, you should know about this threonine to isoleucine pathway because it's end product inhibition. What is the feature of this pathway? Oh, well, it gives it to us. So pathway is related to positive feedback. Remember, no. Negative feedback. Isoleucine is competitive inhibitor. Again, not true. Isoleucine is a substrate for threonine deaminase. That is, again, not true. Isoleucine is an allosteric inhibitor. Correct. So yeah, remember, isoleucine is an allosteric inhibitor. It binds to the enzyme at an allosteric site, just an alternative site. Then it regulates the reaction by inhibiting that enzyme. Very interesting. Question 8. What is a common feature of all enzymes? They all react with substrates. They all decrease the rate of reaction. They are all secreted from the cells and vesicles. They all bind to the active site of their substrate. Well, this is pretty easy. It's a free mark. A. I mean, these are just completely wrong based on the definition of an enzyme. C, I guess you could maybe get mixed up with, but you should know that not all enzymes are secreted from cells and vesicles. Generally, if a question uses the term all, right, it's using like a big umbrella term. It's very unlikely that all is going to be the case, right? Except in the the instance of A, where they all react with substrates, except where it's something very obvious like that. Okay, 9. What is a reason that Taq polymerase is a suitable enzyme for use in the PCR, polymerase chain reaction? Okay, so Taq polymerase, you should know, is the enzyme. They essentially the same function as DNA polymerase, right? They just take it from a thermophilic bacteria. So we had to use this enzyme in PCR, 72 degrees C. We use it at this temperature during the PCR to basically replicate these two strands, replicate the strands of DNA. We use Taq polymerase specifically because it's derived from this thermophile, which has a resistance to high temperatures. So this enzyme won't denature until it's very high temperatures, which is why it's able to survive at 72 degrees Celsius, which is what the reaction requires, the optimum of Taq polymerase. So that's the reason we're looking for. So wide range of pH is not true. Higher temperature than most enzymes. That's essentially what we said. So we'll keep a tick on that. Separate two strands of DNA. Again, that's not the role of it. Allows DNA to be replicated without the use of primers. Again, not true. The primers still have to anneal. So B is our answer. What are examples of non-coding DNA? Okay, dominant and recessive alleles. That is not true. Promoters and telomeres. I mean, that is what you by definition there are non-coding DNA, but let's just have a look at the other options. Oncogenes and tumor suppressor genes. Again, this is not non-coding DNA. Introns and exons, while introns are generally basically just by definition on coding DNA. Exons are coding DNA. So our option is B. This table shows mRNA codons for 3 amino acids, which substitution mutation of a base triplet on a DNA strand will lead to the same polypeptide being formed at translation. So substitution mutation of a base triplet on a DNA strand, same polypeptide at translation. Okay, so we're just looking for, so it's basically talking about the degenerate structure of degenerate property of our amino acids. So multiple codons are coding for the same amino acid. So we're just going to look for 2 of them that code for the same amino acid. So yeah, I won't bother just narrating it. It's best if you just do it in your head. And the option is D. Yeah, the option is D. Yeah, okay. Because we're just looking for something that's going to give us the same amino acid. 12. Which is a feature of phloem sieve tube cells? Okay, phloem sieve tube cells, they are pretty specialized, so it should be pretty easy. Oh, oops. Sorry about that. Phloem sieve tube cells, they have numerous chloroplasts. That's not true. No nucleus, that is true. Lignified walls is xylem. No cytoplasm, not true. No nucleus, yeah, that's the one. Against an extra space. How does the Miller-Urey experiment contribute to the explanation of origin of life? Okay. So Miller-Urey one, if you remember, was the experiment in which they essentially proved that organic molecules essentially to do with spontaneous generation, right? And how that occurs. So how phospholipids form protocells in specific lab, that's not true. Organic molecules arise from, yeah, that's essentially what Miller-Urey is about. This is not true. Not true. Okay. Yeah, it's pretty simple. This image shows a group of enveloped viral particles. What is the most likely composition of the structure labeled X? Interesting. So this is our X. It's always going to be blurry. Okay. And what is the most likely composition? Yeah, I mean, look, it's enveloped viral particles. It looks likely to just be the membrane of the host cell. In terms of viral enzymes, the structure is shaped as a membrane around. Yeah. It seems to be like it's a membrane derived from the host cell. You can tell it's not DNA or enzymes. I'm just thinking if it might be viral cell walls, but just the way they're arranged, the shape, it seems to be a membrane derived from the host cell, which actually makes a lot of sense. Okay. 15, the diagram shows the movement of ions that can occur across the membrane of a neuron. From the diagram, what can be deduced about movement of sodium ions? Okay. So we have this diagram here. It's just a sodium-potassium pump. They are actively pumped out, and some re-enter by facilitated diffusion. So we can see they're actively pumped out here by ATP being hydrolyzed to ADP. And we have A plus on the outside of the cell. And then we can see here we have a transport. We have this channel, and we have a channel and Na plus moving down the channel, which is essentially facilitated diffusion. It is facilitated diffusion. So yeah, this is just true. Looking at the other options, I'm trying to see if there's anything you could get stuck on, but it looks like they're all pretty simple. So yeah, it's just A. Okay, question number 16. In multicellular animals, embryonic stem cells have the ability to differentiate in a range of cells with different functions. What's the term to describe cells with this property? So the property of embryonic stem cells. Remember, this is a pure definition when I'm trying to think of how else you could think of it, but you should just know that embryonic stem cells are what we call pluripotent. Yeah, they have a very wide capacity to differentiate. I mean, you know it's not specialized, right? It's one of these three, and you should know that it is pluripotent. Yeah, okay. The serum shows a glycoprotein embedded in the phospholipid bilayer cell membrane. Which feature of this glycoprotein can be deduced from this image? Okay, so A, it is a peripheral protein that allows attachment to the neighboring cells. Well, that doesn't seem too true because we can see it's embedded in the membrane. We can see it has this embedded region. So it doesn't seem to just be a peripheral protein. B, it's a channel protein that provides hydrophilic channels for carbohydrate transport. I'm not seeing this potential as a channel protein here. It doesn't seem to be a channel protein, especially for carbohydrate transport. Yeah. C, it is an integral protein that may be involved in cell recognition. Now, this seems plausible, very plausible, because we can tell it's an integral protein, it's embedded, and its structure seems to be, well, we can infer that. It seems possible that it would have a role in cell recognition. D is a transport protein that increases the permeability of the membrane to glucose. No, that doesn't seem as likely as option C. Remember, for questions like these, when you're deducing something, read all the options because you never know you could see something better, and you're just gonna pick the most likely one. C seems the most likely, so we'll leave it at that. Okay, 18. What activates the tyrosine kinase pathway in a cell following binding to a receptor? So what activates the tyrosine kinase pathway? Again, this is quite basically just what we call deduction based on our prior knowledge. Yeah, if you don't know how to solve this, just review your material, but our answer is insulin. Insulin activates the tyrosine kinase pathway in a cell after it binds to the receptor. Okay, 19. The graph shows the effect of methylation on the expression of MT1E, a gene involved in the control of prostate cancer development. Patients with a reduced expression of this gene are more likely to develop prostate cancer. What are the effects of MT1E methylation? Okay, so we can see the relative expression here of the methylated and unmethylated in this box plot. Okay, so A. So this is methylated, this is unmethylated. A says it reduces the transcription of MT1E, okay, increasing the risk of prostate cancer. Okay, so that's option A. B, increases translation of MT1E, risk of prostate cancer, okay. It reduces replication of MT1E, reducing risk of prostate cancer. Increases chances of mutation in proto-oncogenes, increasing the risk of prostate cancer. Okay, so this isn't too complicated. It's basically just seeing if you can understand gene expression, right? What is gene expression showing us? Remember that what this is showing us in terms of expression is only relevant in terms of transcription. We don't have information based on our translation or replication because those processes are distinct. They're different processes. But from our data, we can infer that it reduces transcription of MT1E, and we know that this is going to increase our risk of prostate cancer, right? Because reduced expression of this gene are more likely to develop prostate cancer. Okay, 20. What is the function of histones? Supercoiling of DNA during binary fission in prokaryotes. Prokaryotes do not have histones, so not true. Synthesis of proteins, not true. Formation of microtubules, that is just not the role. And condensation of DNA is exactly the role of histones. 21. What is the cause of positive phototropism? Increased concentration of auxin on the side of the stem closest to the light, okay. Degradation of auxin on the side of the stem closest to the light, no. Increased concentration of auxin on the side of the stem furthest from the light, yeah. Degradation of auxin side of the stem, no. So yeah, it's just increased auxin on the side first from the light. Remember, you have a stem that's like this, and then your light is like this. If we increase auxin here, this part grows longer. It causes it to grow like that towards the light. Yeah. Not the best illustration, but hopefully you understand. What role does positive feedback play in fruit ripening? Okay, the production of ethylene leads to fruit ripening, which stops the production of ethylene. Okay, interesting. Production of ethylene leads to food ripening, which causes more ethylene production. Well, this is true. It is the production of ethylene that leads to more ethylene production. Let's just look at the other ones. Production of ribulose biphosphate. Yeah, these are just not true. It's to do with ethylene, we know, so we could have narrowed down to these two. But yeah, it actually causes it to produce more ethylene during food ripening instead of stopping the production. Karyogram shows a chromosome abnormality in a human female. What could cause the abnormality to arise? So we have this abnormality here, chromosome 21. We're seeing a triple chromosome there, trisomy 21. Okay, what could cause the abnormality to arise? Okay, failure of homologous chromosomes to separate during gamete formation. Generally, what we know about trisomy 21, this is the one that's going to be correct, but let's just read the others. Chromosome 21 replicating after fertilization, that's not possible. Gene mutation of the mother's chromosomes during cell division, this would not cause an extra chromosome 21. Polyploidy occurring during anaphase 1 or meiosis in both parents. That would not specifically cause what we're seeing in this karyogram here. It would not look like this if it was polyploidy. So our answer is A. Oops. Okay. The diagram shows an organism from the phylum Arthropoda, a dichotomous key was used to classify this arthropod to which group does it belong. And we have this dichotomous key. Okay. And okay. So Arthropoda. Let's just go through it. Antennas pointing forwards, antennas pointing sideways. Yeah, based on just our classification, we know that we're going to go to 3 here because our antennas are going to be pointing sideways for our Arthropoda. So we're going to go to 3. 3, let's see. 1 pair of legs in each body segment, 2 pairs of legs in each body segment. We know that it's going to have 1 pair of legs in each body segment, right? 1 pair is, yeah, a pair is a pair of legs. So we have 1 pair of legs in each body segment, which means it's going to be C. It's a pretty short dichotomous key, actually, but yeah, C. Honestly, I think you could just use the picture if you didn't know about Arthropoda, if you didn't study. They give you a picture. I think you are supposed to use a picture, actually. Interesting. Yeah. Okay. 25. Scientists study differences in the base sequences of a gene found in 5 animal species in order to determine their evolutionary relationships. A cladogram was constructed based on these differences. What can be deduced from this cladogram? So we can see our cladogram here. Species of Phi seems to be branching from there. Okay. Okay. So what can be deduced? There are only 3 clades shown. Not true. Morphological differences between V and C increase with time. V and C. You can't. That's not able to be deduced from a cladogram here. There is only 1 difference between the amino acid sequence of X and Y. Yeah, look. Even though there's only differing here at this point, that's where they diverged. It doesn't mean there's only 1 amino acid sequence difference, right? That's not what it's showing here. Y and Z have a more recent common ancestor than W and X. W and X. W and X. So Y and Z are their ancestors here. W and X ancestor is here. So yeah, I mean, that's true. What is always a consequence of the evaporation of water from mesophyll cells in leaves of a healthy plant? Plasmolysis occurs in mesophyll cells, not a consequence. That photosynthesis stops, that does not happen when water evaporates. Stomata close to reduce transpiration. No. Water moves up the stem in the xylem. Yes. The key here is to look at always a consequence. So always when water is going to evaporate, as a consequence of this evaporating water, a transpiration stream is going to pull up the water in the xylem. So that's always going to happen. The other ones are either dependent on certain the levels of transpiration, the levels of water in the plant, but this is as always. So it's a key thing to look at the wording of the question. Was involuntary peristalsis in the intestine directly controlled by humans? Again, this is just a pure theory question. It's not the endocrine system. It is not the CNS, not the sympathetic nervous system. It is the enteric nervous system, ENS. Yeah. Which cell is a component of the innate immune system? T lymphocyte. Is that innate immune system? Let's keep a bite of that. Phagocyte, lymphocyte, memory cell. These are all part of the immune system, so we can't deduce it based on that. Innate immune system. So which one could it be? So these two are essentially linked in together. I'm just going to go through, if you didn't know which one it was, right, in terms of innate immune system, how you could deduce it, right? So think about how you could group these together. Which of these offer roles that are in conjunction with each other, that are similar? So lymphocyte and memory cells, they are quite interlinked, right? So you would assume them to be part of the same system. And which other one between these two is quite closely linked with the lymphocyte and memory cell? It's our T lymphocyte. They occur in the same chain of processes in the immune response. So just based on that, we can deduce that the phagocyte is a component of a different immune system, different part of the immune system. In this case, the innate immune system. Another way you could know it is just by knowing about the innate immune system. Just trying to offer a way you could solve this question without knowing it. How would the body respond to a rise above normal temperature? Okay, so we know that it's going to be negative feedback when body temperature is above normal. So we can cut that out. Okay, negative feedback, vasodilation of skin, blood vessels. Yes. Yeah. D says shivering. I mean, have you ever shivered in the desert? I don't think so. So, yeah. 31. All right, female grasshoppers have XXX chromosomes and males have XO, signifying a single X chromosome. An X chromosome would be present in only half the male gametes. A recessive mutation is induced by radiation in the X chromosome of a male. In which generation will the effect of this radiation appear? So it's asking for effect of radiation, basically referring to the phenotype. Okay, we're looking for the phenotype. Okay, so let's say we had the parental generation, right? We'll have our normal mother and we'll have our father that has mutated. Okay, so what's going to happen? Well, in our F1, we'll have our female, and our female would have the possibility to receive an allele combination that looks like something like this. Female will look like this. Now, this does contain the mutated allele. However, remember, it is recessive, so it's not going to be showing any phenotypic effects. So this is still okay. Our male will look something like this. It'll take the normal X allele from his mother, and it'll take the no allele, allele from his father. So what does this result as with this allele combination? It means that we have to move on to the F2 generation. So F2 generation, let's say the F1 of this X and then XM and then our XO, they mate. Now what would their offspring include? Their offspring would include an XM female, just like we had before. And some combinations of females. But what we're interested in here is where the effect will occur. And if you see here, we have the possibility of having a no allele and a mutated allele, which means the males will have this combination. XMO. The F2 males will then inherit this mutated X chromosome because they take no allele from their father and they take the mutated allele from their mother. It's going to result in this. So the first generation will be F2 filial 2 males. 32. The number of hummingbird species in South America has increased continuously as shown in the graph. What could be a reason for the increase in the number of hummingbird species in the period labeled Z? So Z is in this period. So it's in 10 to the 10 to the 10 to the 6, and then we have around like 2 here, right? 2 times 10 to the 6. What could be a reason for this increase in number of hummingbirds? Okay, so during this period, what do we know about which could be a reason for an increase here? So food and shelter are plentiful, and there's little inter-specific competition. This was not true. The competition was still very, very large at this period. We would have to go back in time quite a bit for competition to be reduced. Closely related species can coexist in the same niche. Not true as well. Yeah. Again, they would compete with each other, and it would cause further problems. Wide range of unoccupied potential niches increase. Yeah. So this is a great point, and then there are a few limiting factors. It's not true. There's many limiting factors. Okay. Yeah. So during this period, it's still millions of years ago. There is a wide range of unoccupied niches, right? Not all the niches haven't been occupied. Hummingbirds have a very niche role. They're small. They take nectar from plants, something that no other birds do. So it's a very niche role, and then it leads to adaptive radiation, in which it specializes into this role. It comes to the hummingbirds we know and love today. 33. Polyploidy has been a cause of rapid speciation in some plant genera, such as Helianthus, which observation is evidence that it has occurred. So polyploidy, we know what polyploidy is. We know that we can get tetraploids, we can get triploids, we can get all kinds of ploids with polyploidy. So, yeah. Polyploid plant reproduces asexually. No. It occurs due to the error in fertilization. A polyploid plant produces male and female gametes. No. Which is evidence that speciation occurs. Okay, yeah, that's not evidence that speciation has occurred. Neither of these are evidence that speciation has occurred. Fertile offspring are produced when a polyploid plant crosses with a diploid plant. Okay. Fertilization can occur between polyploid individuals. There. That's the one. So, yeah, speciation is when a new species is formed, and in terms of polyploidy, it occurs when we have this new chromosomal count of a certain species forming a new species, and when they can breed with each other, it's it's literally the definition of species. They can interbreed with each other, members of the same species. So they can produce fertile offspring, essentially, and fertilization can occur. That essentially means fertile offspring can be produced, and that shows that speciation has occurred. Yersinia pestis is a bacterium that caused the outbreak of bubonic plague in 14th century. It normally produces ATP in the presence of oxygen, but can still produce ATP if oxygen is absent. Which term describes this characteristic? So it's essentially asking us which term describes a bacteria that can produce ATP even if oxygen is absent. This is just pure knowledge as well, but even if you didn't know, you could eliminate these two. Remember, if you weren't aware, right, just ignore this and look at these. Respiration and aerobic. We know it's not aerobic because oxygen is absent, so it's neither of these two. So we're going to just remove these two. We have these two here. You could just take a guess, but if you knew, it's facultative aerobic. Yeah, that is our answer. 35. What are common features of the of holozoic nutrition and saprotrophic nutrition? Okay. So we know that saprotrophic nutrition, we're looking for this generic phrase, secretion of enzymes to digest food. We always see that with saprotrophic. So that's going to be one of these two, right? Because we know that common features of holozoic and saprotrophic, they both secrete enzymes to digest food. And what else can we see about it? Well, ingestion of food particles, well, not true for either of them, but that's shared. So it's between these two, mostly fungi and bacteria. So A says it's mostly fungi and bacteria, which would be possible for this sapotrophic, because sapotrophic occurs mostly in fungi. But yeah, our answer is C, because it is not mostly both fungi and bacteria. That is grouping two separate things together here. Okay, 36. The preferred temperature ranges for three species of trout that are found together in freshwater lakes in North America are shown. What can be deduced from this information? So lake trout, rainbow trout, brown trout, you can see there are different preferred temperature ranges. Okay. The niches of the three species overlap. Yeah, they do actually seem to overlap. That seems possible. Brown trout have the most specialized niche. No, this is not true. In terms of temperature range, it's not true. Lake trout can avoid competition by living in colder water. That does seem true. It can stay at 6 degrees, then nothing else can stay there, so that's possible. So I and triple I, which is C. 37. Which is a density dependent limiting factor for a kangaroo? Okay, density independent. Okay, sorry, I misspoke. Density independent. So it would not be predation, right? You can see that's pretty clearly not density independent. Climate change again. Eutrophication is not for a kangaroo, right? Eutrophication won't be important. Yeah, it's pretty clearly a forest fire here. Not many other possible options I would see for a density independent limiting factor for specifically a kangaroo. Black walnut secretes the chemical glucose into the soil surrounding its roots. Glucose inhibits cell respiration in other species of plants. Okay, and it's asking us what this demonstrates. Yeah. So remember, this is the opposite of mutualism, right? It's not helping other plants. It's basically taking out the competition. This is not interspecific competition as well, because it is not interspecific by nature. Parasitism, it's not deals to do with the host. This is allelopathy. Yep. What assumption is made when using the Hardy-Weinberg equation for calculating changes in allele frequencies in a population range? Yeah, so Hardy-Weinberg, we always assume the main thing that you should know is that when we use it, we know the population size is going to be very large. We have to assume it, otherwise, because we're doing it in population, right? If we're looking at allele frequencies, the population size has to be large, otherwise it's not possible to really apply the Hardy-Weinberg equation. The other options, yeah, I mean, to be honest, you should know. We should just know that in answer is A. Domestic dogs have evolved from gray wolves. Evidence suggests that the domestication of dogs first occurred around 30,000 years ago, which best describes the evolution giving rise to the domestic dog. So A, the wolf produced offspring in large numbers which underwent natural selection. That doesn't seem very plausible because we're talking about domestication here. Variations in the wolf population that resemble modern dogs favored wolf survival. That's not true, because we still have wolves today that that required the specific characteristics of wolves, but dogs were a result of breeding, artificial breeding. That's exactly what C says, actually. Wolves showing favorable traits were selected for breeding. Favorable traits as in herding sheep, or in some instances being cute. That's a favorable trait. So yeah, dogs are better suited to changes in the natural environment. That's not true. So yeah, it's just C. Remember that humans bred domesticated dogs, and that's what gave rise to the domestic dog. And all right, that's the end. Yeah, it was a decent paper. It's not too challenging. But yeah, that's a lot of talking. Hope you guys enjoyed the video. I understand some of them maybe would like a longer explanation. Oh, wow, I didn't circle this one. Yeah, some of them I just went through them pretty quickly because it's just assumed knowledge. It's just things where if you can figure it out, you can. And some of them I purposely tried to be a bit more vague so you could figure it out yourself exactly how to solve it. But then again, I will also be sharing the paper in the description as soon as I can. So yeah, I hope you guys enjoyed the video. If you did, please leave me a like and subscriber, and in the comments let me know what you'd like me to upload next. It'll probably be a chemistry video next, but I can do more biology if you guys enjoy it. So see you guys in the next one. Hope you enjoyed it, and Rabwise.